here with Rafkat. He's a professor in weed ecology and management, and I understand he came the same year that I arrived at MSU in 2004. So we've both been on the 7th floor of Leon Johnson for a while now. Fabian works in agroecology and sustainable farming. He looks at cropping systems and things like weed and pest management. And he's also been really involved in the climate science group for extension, and I've been talking to people across the state about what climate change means for them, and I really appreciate that as we're developing the Montana climate assessment. So thank you very much for talking. I think you have one of the more provocative titles. It looks like you could probably cover just about anything. So.
Yeah, thank you. Well, first of all, thank you very much for the introduction and for inviting me, and thanks a lot for coming. I'm really excited about the opportunity of sharing with you some of the research we've been doing for the last 3 or 4 years in my lab. And you're right. I mean, when the last, I think, week or so, some friends told me, well, that looks like a weird title. Okay, and the idea is that this comes from a quote that was said around 100 years ago by Henry Mencken, who was a journalist. And the exact quote is a little bit different. For some reason, this is the way people, I think, remember. But it's that for every complex problem, there is an answer that is clear, simple, but wrong. And in a way, I think it reflects some of the way we tend to manage these type of systems. This photo, I took this photo in the Golden Triangle, that would be central northern Montana, where we have a precipitation of around 8 to 10, 11 inches here. So it's an environment, and in order to preserve moisture, farmers basically have to leave in a fallow, so they can capture moisture. And as you can see here, there's almost nothing else but croplands. So in terms of the landscape simplification, I think this is the most extreme simplification that I can think of a landscape. And also in terms of the biological diversity. If you think this way, we have one species every other year. And this represents hundreds of thousands of acres in Montana. And perhaps because we see these systems as rather simple, we tend to think we can manage them with simple solutions. However, what I'm trying to show today is that these are not so simple, and we need to start looking at the complexity of the system if we want to address the correct questions and try to give correct answers. So in my area of interest, of expertise, I work on weeds, and I do research and extension, meaning I go around the state and provide recommendations for farmers in terms of weed management. In terms of how we manage weeds in this phase in the fallow, it's rather simple. If it's a non-till situation, meaning they don't cultivate the soil, it would be just a repeated applications of glyphosate, which is a non-selective herbicide, pretty efficient, relatively cheap, so farmers like it and they use it a lot. Now in the crop phase, it's a little bit more challenging, meaning they have to check which species they have, and then they will be selecting the best herbicides depending on the community of species they have, which is also very small or not diverse community. Maybe they have 4, 5, 6, 7 species. So, and they can even have some web-based tools where they say, okay, I have these weeds, which products do you give me? And they will check based on the price. So you can think that my job as a weed extension specialist is at the Maytag Determined specialist job, which is the most boring job in town. There's not much we can do. However, I think that there are many problems, and there are many shortcomings of using this approach to weed management, like give a prescription of herbicides. Many of the problems are related to environmental and human health issues, non-target impact, reducing the biological diversity of the plant communities, therefore impacting microbial communities, as well as pollinators and other beneficial organisms such as parasitoids, etc. But I'm going to be talking today about one big shortcoming that I think is the one that farmers perceive in the really short term. Because if you tell them, or my experience, if you tell most of the farmer, well, you're not preserving pollinators, they say, well, why do I care? I have wheat. That's not my main major problem. But they really care about this issue of herbicide resistance. And herbicide resistance is the same issue as antibiotic resistance. When we use a product over and over again, we increase the selective pressure, and then we will be selecting for those individuals that can withstand that application. There's a mutation, and then we apply the selective pressure, and we not normally kill those plants. And this is what a farmer will be seeing, a set of dead plants surrounding one individual that was not killed. So something happened to the plant, it's now resistant. They're probably managing, 5,000 acres, so they won't notice that individual or few individuals. And what will happen, if it's, for example, Scotia, it will get mature, produce 100,000 seeds, then it break and tumble across the landscape, and this is the result. One individual plant that is resistant then will be leaving a trail of resistant individual. So you can see that in a few years, we have these escapes of herbicide-resistant individuals, and now the farmer goes, what am I going to do? I can no longer use Roundup to control weeds in my fallow phase. Now, again, if we want to give a simple, and by the way, this is a serious issue. Worldwide, we have 471 cases of herbicide-resistant weeds, a total of 250 species, and with scientists group herbicides by their chemistry. So we have around 26 families of herbicides, and we have found cases of resistance to 43 of those 26 families of herbicides. Okay, around 160 different herbicides. But we can think that this is just another management problem. So what we will do, and this is an approach that is being promoted by the industry as well as by many people in academia, is say, well, you can tan mix your herbicides. So now you have different chemistry, and you will be killing with around the glyphosate all the species, but you will be adding, say, dicamba to control those kochia plants or paraquat or other products. So it is effective in the short term, except that what we are doing is increasing the selective pressure again, right? And what sooner or later happens is that farmers will be selecting for multiple herbicide resistance. These are individuals which are resistant now to 2, 3, 4, 5, and up to 11 different chemistries of herbicides. So now they're really losing the tools. And if you think this is not something that could happen, we have a case here in Montana. In 2006, one product which was called pinoxidine was released in the market. And there were already wild oat plants which were resistant to other chemistry. And we thought, well, this is a new chemistry. We haven't seen any selective pressure of this herbicide. We should be able to control everything. That's what we expected. However, we got phone calls and said we cannot kill our plants. I said that doesn't make any sense because these plants never experienced the selective pressure of pinoxidine. So we actually went there and collected seeds and confirmed that those plants are actually resistant to 5 different families of herbicides. We tested them with 15 different chemistries, and we cannot control them at all. So basically, we were using every single chemistry that a farmer could use in their crop phase, and they can no longer control these plants. I go around Montana and give extension talks on herbicide resistant periodically, and when I show this case, and I give them the list of herbicides they can no longer use. Now, if you think this way, your livelihood depends on controlling those weeds. It's kind of the worst case, scenario you can imagine. I guess it's like, their faces is like, I guess if you go to a physician and say, well, I'm sorry, you got this terrible disease. You can no longer control these weeds. Okay? So it is a major issue we are seeing here in Montana. And By the way, what we notice is that the physiological mechanisms why these plants became resistant is because they basically use the same metabolical pathway that our crops are basically able to withstand the herbicides. That's why you spray and you kill the weeds and not kill the crops. Now the plant weeds, those individuals have the same physiological mechanism. That means that they are not only resistant to everything we have now, but if in the future there's a new herbicide that's going to be released, probably we won't be able to use that herbicide with those biotypes. Again, the worst case scenario. And I think we arrived at this situation because we have not addressed this issue of herbicide resistant for what it really is. And it's basically an eco-evolutionary problem. It's not just a management problem. And there's quite a lot of interest in different areas of science, including medicine, in using this idea of an eco-evolutionary approach to assess our problems. And it took me a while to understand what they mean, because personally think of evolution as something that takes, long time, millions of years, hundreds of millions of years. Now, what we are realizing now is that evolution is something that can happen really fast. For example, the antibiotic resistance, right? I mean, it's something that occurs maybe in a few weeks, in a few days, if you use the same antibiotic over and over again. So it's a quick phenomenon, much quicker than we thought, and it can actually happen, as we are seeing, in our crop fields. Now, when a farmer gets a problem of herbicide resistance, what they do is basically they will be modifying the ecology of the system. Why is that? Well, for example, they can no longer kill their weeds with herbicides, so they will be going back to a mechanical control practice, tillage. That has a lot of environmental issues associated with that, such as releasing greenhouse gases, losing soil moisture, soil erosion, many problems. But basically, they will be totally changing the ecology of the system, and by doing that, they are creating a new set of selective pressures. And that new selective pressures will change the communities and the genetic composition of those weeds. And this is an example. I don't know if you can see it from the back, but it's the emergence of two biotypes of giant ragweed that a friend of mine sent me this graph from Illinois. He was called by an organic farmer saying, I can no longer control my giant ragweed weeds. So he collected seeds from a close by riverbank and within the field, and he noticed that the weeds in the riverbank in blue, they emerged very early on. While the weeds within the crop fields, they have this in red, they have this long tail of emergence. And that's because farmers use repeated cultivation up to a point where the crop is too tall, they can no longer cultivate the crop, and then the weeds can escape that selective pressure. So the farmer will be maybe modifying their cropping system by putting a new crop, again, feeding back into this idea of evolution and ecology. But very important is that all these processes are occurring with an regional and landscape complexity of the system. And we tend to think of weed management as something that happens in a field, right? You have this weed, apply this herbicide. But actually, there's a huge number of propagules that move within fields and across regions, like I said, from these, kochia plants that tumble across the landscape, but also depends on our management practices. For example, we can have a combined harvester that, cuts wheat all the way from Kansas going to Alberta. So there's going to be a lot of seeds that are going to be moving north and south. So perhaps a simple solution to avoid this movement of seeds into Montana will be to build a beautiful wall. It will work. So anyways, you can see that these problems are not that simple and it requires complex solutions. So now we're faced with a question, what do I do to manage these multiple herbicide-resistant weeds? I'm going to call them MHR, multiple herbicide resistance. And this is a project that Erin Burns did it for her PhD, and we did it together with Erin Lemhoff, Barbara Keith, and Bill Dyer. So the main goal of this project was to try to see if we can use now our crops as the management tool. Can we enhance the competitive ability of the crops so that we can suppress the weeds, because we no longer have herbicides during the crop phase. We still have the glyphosate, but it's only one every other year. So we thought we could be using two types of stresses. One will be an environmental stress, an increased rate of nitrogen from a low level of nitrogen to a little more higher what the farmer will normally use, and one biological stress, which having a crop more competitive. This is seeding at the normal seeding rate or seeding at almost twice the seeding rate, which is something that organic farmers tend to use. So we took a lot of measurements, such as the level of nitrogen in the soil. We checked how many wild seeds were there in the seed bank. And then early in the fall, we selected these areas where we put 1,000 seeds per square meter of either a susceptible line, I'm going to call HS1, or the resistant lines, that's going to call HR3 or 4 for multiple herbicide resistant M4. Okay, so we created these seed banks that we put a lot of seeds to somehow simulate an escape. You know, farmer have a problem of herbicide resistant, a lot of seeds went into the seed bank. What does he do? Then we put these seed bags with soil to check for seed decay because we wanted to see how many seeds just simply disappear due to the presence of microbes occurring in the soil. And then the next spring, we put at random several rings and we check for the emergence of seedlings. We mark those seedlings. Then we sprayed herbicides because we wanted to simulate what a farmer will be doing, and hopefully we will be killing the resistant individuals and the susceptible ones, and the resistant will be allowed to survive if they were resistant. We check for those individuals during the growing season, up to the time where they produce seeds, and from other studies we have estimates of the amount of seed predation in the same area by invertebrates as well as by vertebrates. We conducted this study over 3 years, and we have 4 replications per year. So you can see we have quite a lot of environmental variability. And just to show you some of the results, this is the case of the wild seeds that are being produced per square meter in the spring wheat at the low seeding rate, so the not very competitive environment, at the 4 nitrogen levels. And you can see in blue is the susceptible line, where basically no seeds being produced because we killed them with a herbicide, as expected. But the resistant line, the 3 in yellow and 4 in light blue, produce anywhere between 1,000 seeds to around 11,000 seeds per square meter. Now, the interesting pattern is you can see that as we increase nitrogen, the number of seeds decreased. When we increased the seeding rate, what we saw that effect was even larger. Now, the interesting value here is 1,000 seeds per square meter, because that's the initial population we have. Anything above suggests that population will be increasing, anything below decreases. And you can see the numbers are almost everywhere. 2014 was a little bit drier, and again, quite a lot of variability, but in general, as we increase nitrogen and seeding rate, less seeds were returning to the seed bank. 2015, a good year in terms of seed production, but the same trend of a good approaches to increase nitrogen and seeding rates. So we have quite a lot of demographic numbers. And basically what we can say at this point is like, yeah, in some cases it works to increase nitrogen, in some cases it does not, depends on the environment, depends on the seeding rate. It's difficult. That would be the recommendation. So what we decided to do is to build a stochastic population dynamic model. So we knew the initial seed bank density, 1,000 seeds per square meter. Then we knew how many seedlings emerged the transition to being reproductive adults, the number of seeds that were produced, how many seeds went back to the seed bank in 1 year. And we also were able to estimate the transitions between these stages. So what the model did was basically tell the computer to go and say, set up for one specific management system, say low seeding rate and intermediate nitrogen, and one population, the resistant line 3. Start with 1,000 seeds and choose at random a transition rate. And then you create the seedlings. Then go back and choose at random from those treatments, because we have a combination of years and reps, a transition rate to reproductive adults. And do the same, and you will be moving the population 1 year. And we keep doing that and move the population for 20 years. And we did that exercise for 500 times, so that we can estimate the mean and the variability of what would happen with the population. Another thing that these models allow us to do is to conduct an elasticity analysis. And basically, this is saying, what will happen if I change the transition rate by, say, 10%? I decrease the seedling recruitment by 10%. What will happen after 20 years with that population? And you can check then which is the most, what you can have the highest impact on the population. And then you will focus your management on those particular transitions. So to show some of the results, this is the seed bank density of one load. We started with 1,000 seeds, right? In the low seeding rate, low nitrogen, and you can see that the susceptible line in yellow just disappears because we're killing it with herbicide, right? And the two resistant lines increase, and they go up and down, up and down. And the reason they go up and down is that every time we have a fallow year, and we estimated that the efficacy of the glyphosate is around 95%, and we chose a range of values at random, every time we have that fallow year, there will be some escapes, and those plants produce a lot of seeds, and that's why the population goes up. And then the crops, they can somehow control the population by competing with the weeds. Now, when we did this exercise and we increased the nitrogen for the low seeding rate, you can see that now the population starts to crash faster and faster. And indeed, at the low seeding rate but high nitrogen, we can say we are now starting to control those populations without a herbicide. In the case of a high seeding rate, now we have two stressors, the biological stress, high seeding rate, and the nitrogen, the environmental stress. You can see that the crash of the population is even faster. So now, when I go around and give extension talks and show this result and say, well, if you want to control those populations, you can either, increase your nitrogen at the recommended seeding rates, or you can have a high nitrogen rate, but not really high, but increase your seeding rates. Or if you're really, really concerned, go for the two-pronged approach of a high seeding rate and high nitrogen. But most interestingly is the elasticity analysis, because the elasticity analysis confirms which are the most important management practices that will allow the farmers to reach their goal. The most important or the most sensitive parameter in terms of the elasticity analysis was the density dependent in the seed production. So we really wanted to decrease wheat seed production. How can we do that? Again, by maximizing crop competitive ability, having a high seeding rate, having high nitrogen, or from other studies, perhaps using varieties which are taller and tiller more, so you have a more competitive crop. Also, you can modify your nutrients. The second most sensitive parameter was the efficiency or efficacy, of the glyphosate. Now, farmers, when they see this phase, the fallow year, they don't perceive any benefits, economic benefits. And it's kind of I need to spend money in spraying. I don't see any benefit coming back to me. The benefits come from this phase. So they may be delaying their herbicide application because, well, I can whenever I'm done with my crop. By delaying the application, weeds get larger and the efficacy of the herbicides reduces. So now we're talking about you should be concerned not just of what's happening in your crop, but also in your fallow years, because that's a very important parameter to control your weeds. But very importantly, they need to start thinking about trade-offs, because they need to check out their pockets and see how much money they want to invest in seeds, how much money they want to invest in nitrogen, how much time they want to invest in managing these fallow fields. Again, they don't perceive any economic return. And I showed these results, I think 2 weeks ago. I think it was in Jordan, Montana, and one farmer said, you know what, actually I was trying something similar, but I was increasing seed rate and putting nitrogen, but I didn't get any feeling of the heads of the wheat. And that's because he didn't have enough moisture. Okay, so again, this is an environmentally dependent management approach. And the third most important parameter was the survivalship or the survival rate of those seeds in the seed bank. So now it's talking about the management practices they can use will be to increase the mortality of those seeds, again by having more seed predators, something that we are not even considering here in Montana. But when I was in Michigan and Iowa before coming here, that was a talk that farmers started to have around 15 years ago. And the second issue will be to increase the seed decay. And that's something we really got very excited, because that's something farmers may actually do. They can actually modify by their management practices the soil microbial community, and by modifying their soil microbial community, changing the decay rates of those wheat seeds. And this is one study we conducted a few years ago, where we went and selected 4 pairs of farms, 4 organic and 4 conventional farms that were close by to one each other, and we sampled the soil. We brought them back to the lab and checked for the presence of microbes and the microbial communities. And Sue Pellegrini, she just published a paper where she showed that cropping systems significantly impact the microbial community. And you can see here in green, you have all the conventional farm groups, and in blue, you have the organic farms group. They have a totally different set of microbes. Why is that important? Well, another of our students, Stephen Johnson, what he did a greenhouse study where he was inoculating pots with pea soil that he collected. So just adding a tiny, like a teaspoon of soil into the pots, and he was checking how plants grew with those inoculums. And what he did as a control, he sterilized the inoculum, and when he saw that, when you sterilize the inoculum, this is wheat biomass in here as a function of the different treatments. The first graph here is a sterile inoculum, no difference between the organic and conventional farms, but when you have an active inoculum, the wheat grew much better, but you can see it grew significantly larger in terms of the biomass when you have the inoculum coming from the organic farm. And also, we tested that in competition with different wheat species, such as wild oat and redwood pig wheat, and you say the same result. Not only the wheat grew better when you have the more diverse inoculum coming from an organic farm, but also was more competitive, which brings a lot of hypotheses of why this is occurring. And it was, one of the hypotheses that is that those microbial communities allow plants to forage resources in different pools within the soil. So there's a lot of interest there, and one of the things we are currently doing now is seeing how those microbial communities and plant soil feedbacks change as a function of management systems, as well as different predicted climate scenarios. So we have systems where we increase the temperature and decrease the moisture, and we manage them conventionally, organically, and also we have an organic system where we integrate sheep. So we are very excited. We're getting really interesting results, and we're going to solve a lot of problems. Except that in the first half of my presentation, I was only using my wheat scientist hat. And one thing that I learned when I was working in Michigan State is that I was working there in the entomology department, is that you also need to talk with entomologists because problems get to be a little bit complex there. Because if you ask an entomologist, like David Weaver, he will say, the main problem in these systems is this one, the wheatstem sawfly. And the wheatstem sawfly basically is an insect that has one life cycle per year. The females fly for only a week or so, and then there's oviposits in the best wheat stems, and they spend the rest of the year inside the stems. The larvae will burrow the stems, and that will create around a 20% yield loss. But the problem is that it creates a diapause change, and the wheat stems break, and what you will see is this. Basically the wheat stems lodge and the plants fall, so the farmers have many issues related to harvesting this field. So it's a major problem in Montana. The problem is that weeds and insects don't occur in isolation. So farmers try to manage both of them, but also there's many biological interactions between these two species. And now we work with a different species, which was cheatgrass, Bromus vectorum, or downy brome. And the reason we chose the species is because the wheatstem sawfly can actually oviposit on the cheatgrass. They can also oviposit in wild oat, but we choose to work with cheatgrass. So they oviposit in cheatgrass, and they can reproduce, and they also have a selective behavior. They prefer cheatgrass versus wheat. For the wheatstem sawfly, there's not a good viable management option. So what we have are cultivar varieties. So farmers, when they have a problem with wheatstem sawfly, they select a solid stem variety. So the larvae cannot burrow there and they die. But those varieties are not very competitive for the weeds. They are not really high yielding. So the solution will be to increasing rate, right? That's what I would be saying if I have my wheat scientist hat. It's good for the wheat management. The problem is that when you increase the seeding rate, the solidness of the stems disappear, so it's no longer good for the weeds, it's no longer good for the insects. And also, remember, this is a dry land situation. Moisture stress is a major problem, and you increase the moisture stresses, and chances of having pathogens also could increase. So as you can see, this is now we have a problem that is way more complex than we thought at the very beginning. We can no longer start thinking, okay, have a weed that will spray, have an insect that would just a solid stem varieties. So the question is, how do we manage these multiple pests that occur at different trophic levels and they interact with one each other? And this is a study that Eli Karen did it, and I co-advising with Jim Robinson Cox from the Stat Department, and also David Weaver and Alan Dyer, not Dyer, sorry, I was a member of his committee. So what we did for this study, we selected 3 farmers' fields across Montana, and it's inside of each field, we established 3 varieties: a sawfly-resistant variety, a drought-tolerant variety, and a high-yielding variety. And then we established them at 3 cropping densities: a low, a medium, and a high seeding rate. So we're increasing the margin for weeds in here, right? More high in seeding rate density, theoretically better for weeds. But also we were increasing the cheatgrass pressure by either spraying 0.x of 8 of the herbicide, so quite a lot of control, 0.4 of the herbicides, we expect to see some escapes of the weeds, or 0.2. So we expect to have a very weedy environment. And because nothing really interesting happening in the high yielding variety, it really performs good when there are no pests. I'm going to show results for these 2 varieties, the sawfly-resistant variety and the drought-tolerant variety. Now remember, the sawfly-resistant variety, when you increase your seeding rate, loses the solidness. It's no longer good for the wheatstem sawfly. So we established the fields, again we spray, and then we established a series of rings, 5 rings, where we're measuring the presence of cheatgrass. Then we harvest 2 of the 3 rings to get some estimate of the biome of cheatgrass, so we were able to estimate crop width competition. And at the end of the summer, we harvest the other 3 rings, and we brought them back to the lab, and we separated the cheatgrass from the wheat. We checked for the presence of a larvae or a parasitoid of the wheat stem softline, and we also checked for yields. So the question that came now was, how do we combine all this information to make a multi-pest management recommendation? Because now we have recommendation in terms of herbicides, for insects. How do we combine that? I thought it was going to be easy, but Eli was really smart, and he was a student in the stats department, provided a totally different perspective. He was saying, well, you can't really do that, because for each relationship, you will be using a different statistical tool. For example, you may be doing between cheatgrass and yield, you do a nonlinear regression. Between the width and softline and yield, you do another type of regression, maybe a multiple regression. So you will be using different statistical tools. And how you combine those results, how you propagate errors there, it's going to be extremely difficult. But most interestingly, if we do this, we will be able to just test for direct relationship. Like if we have a solid stem, we'll be decreasing width and softline. And also we have a certain competitive ability against cheatgrass. But there are some indirect relationships there. For example, the solid stem varieties, by competing differently with cheatgrass, will be then changing the ovipository behavior of the wheat stem softline. So there's all these indirect interactions, and we didn't know how to incorporate that. So what Eli decided to do was to combine a structural equation modeling approach with an annihilation approach and use a Bayesian statistical inference. So for the structural equation, it's basically something like a path analysis where you set up a series of relationships and you solve them all together at the same time. For the annihilation approach, what he said is, okay, let's assume we have 3 variables here, x1, x2, and a response variable y. For example, x1 will be a function of x1 and an error term, y will be a function of x1 and x2. Now, x2 is in both sides, in both equations, so you can replace, and by replacing and moving some terms around, he came with what he calls the annihilating model that will be taking into account these indirect interactions. And this is the model we designed, where we have the impact of management on the wheat stem softline, the cheatgrass, and the yield, as well as the indirect interactions. And he represents those indirect interactions with this set of equations. And if you have any question relating to these equations, I have a line in my speed dial, and you can call him. But the way to solve this structural equation modeling with this indirect interaction, he decided not to do it with traditional, parametric, approach, like a traditional stats where you have a certain test and you reject or fail to reject your test. Basically, he was using a Bayesian approach where you construct a prior probability based on your knowledge of the system, run the test, and you increase the knowledge of the system. You have a posterior probability. So he decided to do that because it allows the incorporation of this total effect and pre-existing knowledge and test-specific hypothesis. I'm going to show you an example of how we did that. For example, in terms of the oviposition behavior of the wheat stem softline. We knew from previous studies, our previous knowledge, is that the wheat stem softline actually prefers to oviposit on cheatgrass and survives more on those cheatgrass plants. So, for example, if we have a grain yield per head and a distribution of yields, a normal distribution, we know that the wheat stem softline prefers those large stems. So there will be a threshold value where they will no longer oviposit because they prefer to go to the big yields, the big stems. So in the absence of cheatgrass, we will then be having mean yield of the uninfested heads. Now, when we have cheatgrass, there's competition there. The yields will have now a different frequency of distribution, and there's two different behaviors that the wisdom software could be having. It could be using exactly the same threshold value, and we'll have the same mean yield, or it could start going to the smaller stems because there are not so many good wheat stems around, and the mean yield of the uninfested varieties will be reduced. So in terms of the results, this kind of complex graph is in terms of the seeding rate, the low, medium, and high seeding rate, and the three different herbicides. This is the gray is the low herbicide, so high weed pressure. And as you go darker, we put more herbicides, so the less weed pressure. And this is the mean yield for the sulfite tolerant and the drought tolerant variety. And to summarize this sort of a complex graph, what we see is that in 5 of the 6 cases, the sulfites start to oviposit in the smaller stems, because we see that the yield of the uninfected stems decreases as we have more weeds. So there's a change in the behavior. We can now understand that there's a change in the behavior of the insect because of the presence of the cheatgrass. But at the end of the day, we want to make a management recommendation for farmers. So in a weed-free environment, there's no cheatgrass, and we have here the drought-tolerant variety and the sulfite-tolerant variety. For the seeding grade will be the low, medium, and high. Okay. Without any weeds, the best recommendation will be to use a sulfite-tolerant variety at a low seeding rate, because you have around a 12% increase in yield. And the reason for that is because you have no weeds, you want to have no competition among the wheat, so you preserve that solidness of the stems, and you're controlling the weed sulfite. Right? It's a one pest, one management recommendation. The problem starts what happened when we increased the cheatgrass pressure. So we have a herbicide application of 0.4. We have some weeds there. We can think, well, let's go for the solid stem variety and increase the seeding rate. Right? That would be the logical, because you will be controlling both species. But remember, you lose the solidness of the stems if you do that. And indeed, the best recommendation will be to go for the, drought-tolerant variety at a high seeding rate, because the solid stem variety doesn't do that good. So the management recommendation will be, for a farmer, you need to change the varieties, seed for a high seeding rate, control your weeds, and then go back and take care of your weed stem sulfite. Why can you do that? Again, because there are trade-offs. You cannot manage both pests at the same time in the same year. You need to start planning ahead. Similar to what we have with the wood fallow. Farmers need to start looking at the fallow as a management phase for what the weeds they have in their wheat year. So what would happen in a weedy environment? We have a lot of weeds. Well, in that situation, basically, there's no difference across management recommendations, which basically says that weeds rule. So, at the end of the day, we rule. So, I hope that these 45 minutes, I was able to show you that what it looks to be a very simple system, and not that simple. There are very complex interactions occurring there, and we can no longer go with these simple management solutions, because at the end of the day, they will be wrong. They will start to backfire on you. So, when we do that, we need to take into account the complexity of the systems, as well as the need to get trade-offs. You can't get the best of both worlds at the same time. We do that by, again, going back to these simple systems and trying to understand the complexity of them, and always become to be amazed by how complex they are, even that they don't look like that. And of course, this is not research that I did by myself. It was done with a lot of students and researchers and helps, and including some sheep, because we're grazing cover crops. So a lot of help from many people, particularly many undergrads who usually don't get recognized. And with that, I think we have like a few minutes for questions, right?
That's great.
Okay,
Yeah,
thank you.
What's the mechanism or mechanisms by which added nitrogen reduces the survival of weeds? Is it a
Yeah, so, okay, the question, if I understand correctly, is what's the mechanism by which adding nitrogen reduces the survival rate? So the mechanisms could be said is because you create a more competitive crop, because and then there's basically sort of an asymmetric competition between plants. Now we were scratching ourselves. It's like what happened? What's the second mechanism behind that mechanism? And we were looking for a lot of references, and I think what happens is that we have a delay in emergence. We don't really understand why, but we saw, and it's years consistently, we saw a delay in the emergence of the wild oats in comparison with the wheat. So by having those wheat emerging and growing faster, I don't know if the word exactly is delay in emergence or faster growth at the early stages. By allowing that process to cure, probably what you're doing is you are enhancing that asymmetric competition between the crops and the weeds. Okay.
could differentially induce limitation by another resource?
Sorry?
Could induce limitation by another resource?
Oh, totally. I mean, that's how I tried to show in that very last slide. I mean, and I said in my recommendation, when I showed this to a farmer 2 weeks ago, the limitation was moisture in his case. Definitely. And not to talk about, I mean, I mentioned the issues of environmental costs of increasing nitrogen. We don't even, even touching all the environmental fronts of increasing nitrogen, from ranging from greenhouse gas emission to leaching. That's, oh yeah, most definitely.
How was the nitrogen placed? I mean, did both the wild oats
It
and
was broadcast.
was broadcast, so they both had equal access?
Yeah, so the
Just
question was,
before
how
it
was the
came out.
nitrogen placed, was broadcast?
Right after we seeded the wheat.
Yeah, so it was broadcasting, and one another approach which will be perhaps much easier, but we decided not to do it, will be to ban the nitrogen. So that will be perhaps more effective in terms of not needing to increase the nitrogen and having all the associated problems and have a similar result.
So wonderful.
Okay.
Now, if I just tried brainstorming, when they were having trouble with Drosophila in the orange orchards, they tried to attack the pest itself. Is there any way you could introduce decay susceptibility into the weed seeds genetically? Is there any way you can modify the problem, the genetics of the problem of the invader? So I don't have a proposal, but I'm just
For the chicas, you see?
yeah. Well, or the Sarcobatus, not Sarcobatus, the weed. Is there a way to introduce genes into that population to its detriment? And those should be selected against. But I don't know.
I don't know. I have no clue. One thing that, not exactly related to what you're saying, but one of the very first things that we thought is that there should be some ecological fitness cost of the resistance. Because, plants, like any organisms, you have a certain amount of resources. You either reproduce and grow, or you defend yourself. So if plants are investing so many resources in defending themselves against all these 15 different herbicides, they should be, we thought, a fitness cost associated with that. Zero. None. I mean, almost nothing. So there was a little bit in terms of seed production and tillering, but not a clear fitness cost. So, like we thought, okay, we're going to be naming them down with pathogens because they won't be able to defend, and there will be a huge decrease in the seed bank. That's what we thought. No, that was not a solution. So,
If there's no fitness cost, why,
sorry.
if there's no fitness cost to the resistance, why isn't it really highly frequent to begin with?
If there's no fitness cost, why it is frequent?
Why is it not frequent to begin with? In other words, why did they even develop the herbicide if it wasn't effective? I mean, if there's no fitness cost, it seems like that mutation should be
So,
very common.
First, to my knowledge, the idea of fitness cost was proposed in tons of studies and has been only, successfully shown in very few cases. Very, very few cases. Now, resistant individuals tend to spread relatively fast in the environment. This population, this specific population that you said, is not present because luckily, I didn't tell the end of the story, the farmer called us really quickly and we said, like, be careful, they're just terminate the crops immediately. He was smart enough, he killed all the crop, sacrificed the crop, and killed all the weeds, and to our knowledge, it didn't appear again. Except that a few years later, we found a very similar case in Manhattan. So I doubt there's any equipment going from the Fairfield Bench area where this mud barley to Manhattan was basically potato. So I think it's just natural occurring selection. And, again, that biotype was early controlled. So, but in the case of kochia, it was found 1 year, 50 miles north of the border in Canada. And by the way, I read an article where people were saying they need to create a fence. So someone was actually proposing that, so build the wall. So, and it took just 1 year to be found here in Montana, and 1 more year to be found with multiple herbicide resistant to glyphosate and ALS, and I'm pretty sure it's going to get to growth later. So, any questions?
Great,
Thank you.
thanks very much.